Now, let me introduce you our speaker today, Gabriel Gomila, Group Leader of the Nanoscale Bioelectrical Characterization Group of IBEC. Gabriel Gomila got a PhD in physics from the University of Barcelona in 1997. Later on, he was postdoctoral researcher at 3 different universities in Italy, France, and Spain, where he specialized in the theoretical modeling of nanoscale electronic devices. In 2001, he moved to the Department of Electronics at the University of Barcelona, thanks to a Ramon y Cajal Fellowship, where he expanded his research interest towards the merge of electronics and biological fields. In 2005, he became Associate Professor at the University of Barcelona, and in 2008, Group Leader at IBEC. Since 2017, he is full professor at the Department of Electronics of the University of Barcelona. In 2014, he was awarded with the ICRE Academia Prize for Research Excellence. His current research interests are centered on the understanding of the bioelectrical phenomena at the nanoscale. So, Gabriel, thank you very much for being here today, and the floor is yours. Thank you.
Thank you very much, Teresa, for the presentation. Today I'm going to speak about the research that our group is doing on the field of bioelectricity, and specifically on the new technologies for bioelectricity. So before entering to the activities that we do, I will just do a sort of introduction to the main concepts of bioelectricity. So what is bioelectricity? In a general way, we can define the study of electrical phenomena that takes place in biological systems. Probably you are familiar with processes like electrostimulation, in which when you apply a current or a voltage to a living organism, then you can elicit a response from that organism. Also, probably you are familiar with the recording of bioelectrical signal generated by the body, like for instance the electrocardiogram, where you record the electrical activity generated by the body. So the first thing is to understand what are the main players in bioelectricity. So if we consider a cell sitting in some extracellular medium, then the cell is a quite heterogeneous system, and the separation between the cell and the extracellular medium takes place through a very thin layer, which is the cell membrane, which is only 5 nanometers thick. This cell membrane separates the inside of the cell from the outside of the cell. Then this membrane is not permeable to ions in general. However, inserted in the membrane, you can find some specialized proteins, which are called ion channels, which when they are open, they allow ions to flow from one side of the cell to the outside of the cell. Then normally ion channels work under passive condition, which means that since the concentration of ions inside the cell and the concentration of ions outside of the cell is not the same, when the channel opens just by simple diffusion, ions flow from one side to the other. In order to keep the balance of ionic concentration, there are other specialized proteins, which are called transporters, which are able to pass ions from the one side of the cell membrane to the other side, but this time against the concentration gradient. For this reason, these transporters need energy, which is the energy that we consume in our bioelectrical processes. So these are the main players in bioelectricity: ions, ion channels, transporters, and cell membranes. If we want to put this into physical magnitudes, what are the physical magnitudes that are normally considered in bioelectricity? Well, basically, these are the concentration of ions, which are specified in the outside, in the inside of the cell, and also at the cell membrane. The conductivities, which explains how easy an ions move in response to electric field. Again, different conductivities inside, outside, and at the cell membrane. And then there is another magnitude, which is probably people is less familiar with, which is the dielectric constant, which is the measure. Here I have made an insert on the left of the screen. What is the dielectric constant? So the dielectric constant explains how the cell membrane polarizes in response to an external electric field. So the cell membrane has small dipoles. When there is an electric field, this dipole orientate, and how much this orientation takes place is proportional to the dielectric constant. So again, there are different dielectric constants for the inside of the cell, the outside, and the cell membrane. In this talk, I'm going to speak a lot about this dielectric constant because this is one of the magnitudes that we measure. Apart from this, there are ionic currents, they are electrical potentials, and all of them in general are space dependent, time dependent, and more importantly, they are voltage dependent or dependent on some ligands. So this is typically the physical description that we do for bioelectricity. Another thing that we have to take into account that there are two types of bioelectricity, what is called passive and active response. So let's just start from the right side, which is the passive response. In this case, it just the cell is just considered to be a material, and then we apply an external electric field by means of a source. And for instance, we measure the current that crosses the cell, and this gives information about the physical magnitudes, the passive properties like the conductivity, the electric constant, etc. But the most interesting part normally is the active response of the cell, in which the cell itself is generating the electrical currents. As you see in this electrical circuit, there is no source here. There is only a meter because the source is the cell itself, which is the one that generates the electrical currents into the system. Normally, it can be some confusion because to produce an active response, you can do it by using chemicals, or you can do it by using electrical stimulations. So then sometimes you see also a source in this circuit, but this source is used to stimulate the cell. But normally you don't need it if you don't want to use electrical stimulation. In our work, we have concentrated a lot more on this part, although recently also we work in the active response of cells. Okay, so in bioelectricity, technology has been an important driver, and the discovery of new tools and new methods has allowed us to understand better bioelectrical phenomena. Typically, we need, from one side, we need sources to stimulate the cells or to perform passive studies. In the old times, they were just simple apparatus. Nowadays, they are just source meter with a lot of electronics inside and some modern microelectrodes. These are the ones that we use to stimulate cells in bioelectricity. Also, you need meters, so devices that record the electrical activity or the electrical response of the cell. Again, in the past, there were relatively simple apparatus that with the invention of the amplifier, this was a big step in the recording of signals. Then they transformed it again into modern meters coupled to computers to acquire and process the data. And finally, also microscopes play a very important role in bioelectricity because they help us to see the structures that we are studying. In the old times, the optical microscopes or the beginning, the staining method used to visualize, for instance, neurons, nowadays are substituted by powerful optical microscopes with a lot of modern fluorescence labeling. With all these developments, nowadays, the typical setup in bioelectricity is composed of an optical microscope connected to some microelectrodes, which can be positioned very accurately. The electrode is positioned on the cell that you want to record, for instance, and by using some meters, you can record the electrical activity of the cell. In some cases, instead of using these sharp electrodes, you can also use planar microelectrodes in which the cells sit on top of them, and then you can use this to record the electrical activity. This is just to give an idea what is the standard setup in bioelectricity so that we can see better the difference with the technologies that we develop. So typically, what are the measured quantities in bioelectricity? So there are basically two electrical potentials, which can be, for instance, corresponding to the active response of the cell, for instance, as the famous action potential, or they can correspond to just the passive response of the cell. And the other thing, in this case, the recordings are done intracellularly. It means that one of the electrodes is located inside the cell. Or you can measure the electrical activity by measuring what is called extracellular field potentials, which is the electrical activity outside of the cell, which is what we do in the electrocardiogram, for instance. Besides current potential, you can also measure currents. And for instance, you can measure transmembrane currents across the surface of the cell. You can also measure alternating currents. This is the one that are more connected to our work. In this case, you can obtain information on the complex permittivity of the cell, or if you have even better setup, you can even measure single ion channels, the current flowing through one of these single molecules. So basically, this is the standard bioelectricity. So what is the activity of our group? Well, our group has devoted some years to develop new technologies for bioelectricity. Especially, we focused most of the time in passive properties of cells. And also, our focus has been to go from the larger scale to the very small scale in order to investigate properties that until now could not be investigated. So to start with, I will start with some old stuff, which was done some years ago, but this serves us to introduce the main concepts. So let's imagine that we want to measure the passive electrical properties of the cell. One setup that we can use is what is called the impedance flow cytometer. In this case, you have a microfluidic channel in which the cells are flowing through this channel. And then on the bottom of the channel, you have some electrodes. And then you put an alternating voltage between pairs of electrodes. Then when the cell is passing by between two of these electrodes, it modifies the electric field in this region. And this physically implies a modification of what is called the impedance between these two electrodes. Therefore, by measuring the impedance between the electrodes, you can measure, in some sense, the passive electrical properties of the cell. This is the mechanism which is used in the culture counter, which is a device used to count cells in clinical hospitals. But here we use an alternating current to measure these properties. So this is one of the setups we developed. Basically, you have these different branches of our microfluidic system in order to align the cells one by one. We put pairs of electrodes, and then when the cell passes by the measuring electrodes, we can record the presence of the cell and eventually also the properties of the cell. This is just a picture of one of the devices, which is a few millimeters or a couple of centimeters in size, just to show that this is a small device. Here you have a small video. Here you have the channel. We are going to see the particles passing by here, and here you have the recording electrodes. And at the bottom, you have the electrical signal, which is recorded. So initially, there is nothing because there is no cell. And later on, when the cell is passing by, in principle, this should work, but it doesn't work in my screen now. But in principle, you should see a cell passing into this channel. And when the cell is here, you see this spike. And when a second cell passes by, there is a second spike appearing here. And this is just to show in a practical demonstration that cells can be detected in these type of channels. Here you have the video. You see one cell passing by, and now another cell will come, and you will see it passing by. You see we can detect. And you can count up to 1,000 cells per second if you want. So a typical experiment, for instance, of this system is, for instance, to take a mixture between red blood cells and white blood cells. And then when you use this apparatus, you can distinguish the amplitude of the signal for the white blood cells is much larger than from the red blood cells. And in this case, the reason is because white blood cells are bigger in size as compared to red blood cells. So now the question is, why the signal depends on size? And the second question is this signal depending on the electrical properties of the cell, or it doesn't depend? So to answer this question, we need to do some mathematical analysis of the electric fields in the cell. We take a very simple model in which the cell is modeled like a sphere, where you have a membrane which is very thin, and the inside of the cell, which is the cytoplasm, and the outside of the cell, which is the electrolyte solution. In here, when you do the mathematical calculation, if you calculate the current that flows through these cells as a response of the voltage, depending on the frequency of the voltage, the impedance that you measure has a certain dependence on the frequency. When there is no cell, is the dashed line here. Practically, you see a flat response, and then at some point you see a decaying when you go to very high frequencies. Instead, when the cell is between the electrodes, you see again a flat part, but then you see some structure here. And this structure here depends on the electrical properties of the cell. So the first bending depends on the properties of the membrane, the second one on the inside conductivity, and the third part on the dielectric constant of the inside part of the cell. And the first part depends on the size. That is, the separation between this line and this line depends on the size of the cell. In the previous experiments, they were done at 120 kilohertz, so it means that we were sensitive only to the size. And why are we sensitive to the size? Well, the reason is because basically, in these experiments, the conductivity of the cell membrane can be assumed to be very low, so ideally zero. So the cell is an insulator, basically, and then the field lines try to avoid it, and then how much they avoid the cell depends on the size of the cell. If we want to know more about the electrical properties of the cell, you need to go to higher frequencies. We have not done this, but other researchers have done. I just show example from another group in which they have done experiments at much higher frequencies, and they have seen that indeed they can distinguish, for instance, the properties of the cytoplasm or the properties of the dielectric constant of also the cytoplasm by measuring at higher frequencies. So in principle, you can detect the passive electrical properties of cells by using these type of setups. However, what happens? Well, it happens that in reality, cells are not homogeneous. So they are highly heterogeneous. You see there is a strong of internal structure in the cell. And also the plasma membrane, which is here, is highly heterogeneous. So with the microelectrodes, you cannot detect this heterogeneity. And the second important thing is that besides cells, you have smaller entities, like for instance, bacteria, which are much smaller, even viruses, but which are only 100 nanometers, or even single biomolecules, which are only just 2 nanometers. As long as you make the system smaller, the detection of the electrical properties by using microelectrodes becomes more and more hard. And then at some point, it's not possible anymore to do by using microelectrodes. So this means that you need alternative technologies if you want to address the electrical properties of these small scale systems, or if you want to map the heterogeneity of the electrical properties of cells. And precisely this is what we have been doing in our group. To this end, we have developed a technique which is called scanning dielectric microscopy. This is a technique that we developed together with Laura Fumagalli, which is now a group leader at the University of Manchester. And we worked on this over the last decade, basically. The idea is in principle simple. So we use a sharp electrode, you see, very sharp electrode. And then the idea is that if you use a sharp electrode, then you are sensitive to local properties. This electrode is mounted on what is called an atomic force microscope tip, which is this cantilever, which is inserted in an atomic force microscope, which is a type of microscope which is able to measure at very high spatial resolution. And then you can use this microscope to do two things: to measure the topography of the system, and at the same time to measure the electrical properties at the nanoscale. You can implement this by using current measuring technique, which is called the current sensing mode. And this is what we did at the beginning of our research. But in the later years, we have focused more on what's called electrostatic force sensing. Essentially, the system is very similar, but now, instead of measuring a current, we measure a force. And this force depends on the properties of the sample also. If you put the force into terms of measurable quantities, let's compare. In current measurements, you measure normally the impedance, and the impedance depends on resistance and capacitance. Just to put it briefly, in the case of force sensing, instead of the capacitance, you measure the capacitance gradient, so how the capacitance changes with distance. But at the end of the day, this is the same because the electrical properties are contained in this magnitude. So by using it, you can get the same information as for current sensing, but the advantage is that this is much more sensitive. So then you can get much higher spatial resolution. So how it works, this technique in practice. Let's put a practical example, which is the case of a nanoparticle. You see a very small nanoparticle, only 40 nanometers, much smaller than a virus, for instance. And this is made of a polystyrene, which is an insulator. So with the microscope, the first thing we do is to take an image. This is the topographic image of the nanoparticle. And here you have the surface topography of the nanoparticle. Then you take an electrical image by applying a voltage between the tip and the substrate and scanning the tip close to the sample that you want to measure, in this case, the nanoparticle. And then you record the electrical signature. So this is an electrical property of the particle. This is the topography of the particle. Then we do an intermediate step, which is to calculate the radius of the tip, because this can change a lot during the experiments. So we can do it in situ, and then we get the radius of this very sharp tip. In this case, you see 6 nanometers, very small radius tip again. And then what we do is we combine the geometry of the tip, the geometry of the nanoparticle into a numerical model, and we can calculate the force, the theoretical force acting on the tip. And this force depends on the dielectric properties of the material, in this case, the dielectric constant. So then by varying this parameter, we can try to match the measured force with the theoretical force. And for instance, in this case, if we take the value here, which is around 2.5, you go here, 2.5, you cross in this point, and this is the, for instance, in this case, the dielectric property that you measure. This is 2.4. The nominal value is 2.6, so this is just showing that the technique works. Okay, so we have applied this technology to many different biological systems. I will review some of them. So we started by considering, for instance, single bacterial cells. In single bacterial cells, the same as before. So you have the surface topography of the cell, which is around 1 micron by 2 micron size. This is the electrical signal measured. Then we do all the numbers and we calculate the dielectric properties of different cells. Here we consider Salmonella, E. coli, Listeria sakei, Lactobacillus sakei, and Listeria innocua. And in this case, what we observed is when we measure in the dry conditions, so with no water, all bacteria, this is the gray band, most of them, they show practically the same value. However, when we put some humidity in the ambient, we see that the bacteria, they show 2 different behaviors. Some of them just increase a little bit the dielectric properties. Some of them, they increase a lot the dielectric properties. And for those of you which are familiar in the microbiology, you can identify that this group corresponds to gram-negative bacteria, this group corresponds to gram-positive bacteria. And what happens is that these 2 bacteria, they have, the cell wall is different, and due to the different properties of the cell wall, they get humidity in a very different way. And since humidity has a high dielectric constant, it modifies the dielectric constant of the cell. So by itself, this is not a big discovery, because gram staining was discovered 100 years ago, but it was a good message to say that with these technologies, you can probe properties of single cells that until now were not possible to probe. We have done the same by considering other bacterial systems, like for instance, bacteria and endospores. I don't have time to go there. I just put the reference there if you want to look at. Or we measure also similar properties by considering higher frequencies, as we did in this work, that also I don't have time to go into that. But this just was one step that it is possible to measure the electrical properties of very small scale objects like a single bacteria. Then we move to another property. So we considered the cell membrane. If you remember, I mentioned that the cell membrane is playing a very important role in bioelectricity. So this cell membrane is composed of different components. Basically, it's a composite of lipids, proteins, and cholesterol. So what we have done is we prepared artificial samples that contain lipids, proteins, and cholesterol. And then we applied our technology to this in order to see what are the electrical properties of these different components. And what we have obtained is that for lipids, we obtain a relatively low value, which is around 2, taking into account, for instance, that water has a value of 80. So that's a very small value as compared to water. Then for proteins, we obtain a relatively larger value, which is not exactly the same as for lipids. And cholesterol was sitting in between. So this was a first indication that the dielectric properties of the cell membrane are not homogeneous, as we have assumed at the beginning. They can be very heterogeneous depending on the composition that you have in different parts of the cell. Just a remark, a technical remark, if you look at the scale bar, just this number is extremely small. So Z to farad is 10 to the minus 21. And this is why force sensing techniques are necessary in order to analyze these systems. Okay, so these measurements are done in dry conditions, because in this way you don't have the effect of water. You can study just the property of the material itself. But of course, the important thing is to do the same measurement but in liquid, which is where normally these samples are located. Some years ago, we developed this technique also to be able to work in liquid. It's a little bit much more complicated. In particular, you have to work at high frequencies in the megahertz range. If you analyze the same sample or similar samples, you observe that now the dielectric constant of a membrane increases a little bit with respect to air. And we suppose that this is due to the fact that the cell membranes gets hydrated. If you now consider different compositions, for instance, a pure DOPC or a pure cholesterol-containing membrane, the topography doesn't look that different. But if you go to the electrical signal, it looks extremely different. So this means that cholesterol is modifying the electrical properties of the cell membrane. And in fact, if we put numbers, we observe that for cholesterol-containing membranes, the value is relatively low, very similar to what happens in air. So our conclusion is that maybe cholesterol, the effect it has is to suppress the presence of water inside the cell membrane. You can do similar stuff with more complicated structures, like for instance, liposomes. So liposomes are used a lot in drug delivery applications, and it is a model system, which is a first step towards going to a more realistic system like cells. Here you have the topography of a large number of liposomes on a surface. This is the corresponding electrical image. And then what we have done is to calculate what is the equivalent dielectric property of these liposomes depending on the size. And what we have found is that they group in different branches. So they don't show the same value depending on the size. They depend on the size of the result, but also there are different branches. By looking at a more sophisticated model, we have been able to show that in fact, the different branches correspond to the different numbers of liposomes which are inserted inside the liposome. These are multilamellar liposomes. And that's an interesting result because this shows that with this technique, which is non-invasive, label-free, you can obtain information about the inside of very small scale objects like liposomes. So going still further in size and a smaller size, we have also measured in the past the properties of viruses, which are nowadays very famous, unfortunately. So we consider the bacteriophage, and which is a virus to infect bacterial cells. And basically a virus consists of DNA, strongly packed inside, surrounded by a capside, which is made of proteins, and a tail, which is made of proteins. So we have measured the properties of the different parts, the tail, the capside, and the virus particle. For the virus tail and capside, we obtain more or less the same value, and similar to what we have obtained before for protein samples. But the surprise came for virus particles, because virus particles, they show a much higher dielectric response. Then, if we take into account that a virus is nothing else than a protein and a shell and containing DNA, if you put numbers, you obtain that the DNA is the responsible for this high polarizability of viruses. That was a surprise at that time because normally the value that was assumed in the literature was smaller. And this showed the powerful of using these nanoscale techniques to address the electrical properties of samples. And this number has been verified by atomistic molecular simulations. Also, in a collaboration, so we collaborated with the work done in the group of Laura Fumagalli in order to understand also the properties of water when they are confined in very small channels. This is interesting because in any biological sample there is water attached, and this water is supposed to be strongly confined. And the discovery here was that water has very different properties when it is confined as compared to when it is in the bulk. So these will represent the bulk properties, and these are the confined one. You see there is a strong reduction of the polarizability, and this can have a strong impact also in the biological properties. So the summary so far is that we have made a sort of cartography of the dielectric properties of different samples, lipids, cholesterol, samples containing proteins, samples containing DNA, or complex structure like full viruses and full bacterial cells. And everything, this gives a, a picture of what are the electrical properties of different components in biology. But the next step we wanted to do is to pass from single points to maps, because until now, what I have been showing is the properties of single points, or the equivalent dielectric properties of the system. But now we want to map point by point the electrical properties. So in this sense, we developed what is called the force volume mode for scanning dielectric microscopy, in which we measure at every point of the sample the electrical properties. And the idea is the same as before, to measure a lot of more data than before, to reconstruct some complex images, to use more elaborated models, theoretical models, and then the idea is to get a mapping of the electrical property, not just an average value at the nanoscale. And this is the first result we have obtained. This is a single bacterial cell. This is the surface topography. And now here you see we obtain a map of the dielectric properties. And this is far not homogeneous. In fact, you see very relatively high values here and low values at the borders. And this is related to the composition of the cell membrane, because there is more lipids, which has low dielectric constant in the size of the cell. And in the center of the cell, we expect to have proteins and DNA, basically, inside a cell. Before, what we measured was the value here at the center. So only one value we obtained. Now we obtain the full map. And this is a much more interesting result, because in principle, for instance, we can use it to map the composition, no? Because if we take the table that we derived before, we can make an assignment, and we say, okay, the low values, which are the blue ones, correspond to the lower value here, so it's rich in lipids. The intermediate values probably is richer in proteins, and the central property part is probably rich in DNA. So this is a label-free composition map, which can be of interest in some application. This again thanks to bioelectrical properties. We have done also the same with eukaryotic cells. Initially, we worked with fixed cells, and in the dry condition, the result is similar to what I have shown for the single bacterial cell, but now at a much larger scale. And again, you see there is a distribution of values that is telling you that the electrical properties of a single cell are far from homogeneous. And in particular, you have the region where there is the nucleus, which corresponds to this part, has a larger dielectric response, and probably this is because the nucleic acids are located there. In the borders of the cell, you have the cell membrane, which is rich in lipids, so this is why you get a low value. And in between, even you can see even here some microfibrils probably, which are very rich in proteins, and this is where we can distinguish this in these images. So again, this technology shows that you can do composition maps, for instance, or other studies, but still we have not explored further this. Of course, the more interesting thing will be do the same with living cells. That's far, far more complicated. We are working on this. Still, we have not completed this study, but at least we have been able to do to record electrical image of living cells, which has been complicated. And now we are looking for a more quantitative analysis in order to see if this contrast that we see here, how it is related to the electrical properties of this time of a living cell, which is what we want to do. Probably in some months we can have some results on this. Okay, so in the last part is trying to explain the results that we have obtained in order to integrate the 2 points of view I have shown so far. So macroscale measurements and nanoscale measurements together. And to do this, the device that we are developing is a system that contains microelectrodes, you see here, and which is working together with the scanning dielectric microscope. And everything working under liquid conditions, which are the conditions that are suitable for cell analysis. What device we have developed is in a collaboration, we have integrated what is called an organic field effect transistor. This is a sort of amplifier that amplifies the electrical signals that are recorded by these electrodes. So then that cell will sit in between the tip and the electrode, and then simultaneously we expect, this is not something we have done yet, but we expect in the future to be able to record both the electrical properties of the cell at the macro scale and simultaneously the electrical properties at the nanoscale in order to gain further view. So what we have achieved so far in this progress Well, the first thing is to show that when you have such complicated transistor, still you can work with it with the scanning dielectric microscope. And the trick we have used is to use the tip as what is called the gate. So what is happening in a transistor like this? In a transistor, there is holes, in this case, flowing from the source to the drain in that direction due to application of a potential here. And then the number of electrons that you have here depends on the potential between the source and the electrolyte. If by modifying the voltage in this electrode here, you modify the number of electrons that you have in the channel. And then, for instance, if you record the current flowing in this direction as a function of voltage between in that direction, you see that for some voltages there is low current, but at some point there is a high current increase. It increases 3 orders of magnitude. This is the mechanism of amplification. This is what is used in a transistor, in conventional transistors, to amplify signal. So this system, in principle, is expected to be very sensitive to detect electrical signals that takes place into this part of the sample. So this was one step. So the second step was to show that, okay, now we want to show that if we put the scanning dielectric microscope coupled to this system, still you can run the scanning dielectric microscope. And this is just a demonstration that we have done recently, in which, for instance, you see this is the surface topography of this device here, where you see the source, which is this part, the drain, which is that part, and this is the channel, which is the medium part here. This is the topography, and this is the electrical properties of the channel. You see that the source, the drain, they have different electrical response, and here in the channel you see some structure that reflects the electrical properties. So this is a demonstration that we have done recently, and in which we have shown that indeed you can run a scanning dielectric microscope coupled to such a complicated transistor. Then the next step is to show that this transistor, they can indeed record electrical signals coming from cells. So this is a work that we have done also recently in a collaboration with the group of Marta Mastorens and Nuria Montserrat from IBEC and IGMAC respectively. And the idea here is, well, we have the transistor, which are these black parts, these are the electrodes. And on top of them, you have some cardiomyocytes. In reality, they are human pluripotent stem cells derived cardiomyocytes, but for simplicity, I will call them cardiomyocytes. So there are several of them. This is not a single cardiomyocyte, there are several of them. And the idea is that now this configuration is like this. So you have the cells here, you have the transistor here. Now, instead of putting the tip, we put a wire just to bias the transistor. And now the idea is, if these cells emit some electricity, they will modify the electrical properties of this electrolyte. And this will influence the current passing through the semiconductor. Well, this is a reality, but before that, let's show that this setup is very sensitive. For instance, let's imagine that the electrolyte changes the voltage just 20 microvolts. That's an extremely small electrical change. This is very difficult to measure directly, but if you record the current flowing from source to drain, you see here a measurable quantity which is in nanoamperes. And nanoamperes is something relatively easy to measure. So this means that just by varying the properties, the electrical potential in this electrolyte by 20 microvolts, in principle, you're able to detect by using this integrated transistor into the system. Okay, so that's the theory. Now let's see in practice what happens. So the first thing you do is you take the sample, the system without cells. And this is the baseline. You see there is some noise, but this is because the system is in liquid, is working with several things. But this is a small noise, but clearly this does not represent any special feature. What happens if you put the cells there? So you play the cells, you need some time, so sometimes some days, I would say, so that the cells get well adhered to the surface of the transistor. But once this happens, what you start observing is a sort of beating. You see? This is the typical beating of cardiac cells. And this electrical signal here, in some sense, represents the electrocardiogram of these cells. This is the same as we do when we record an electrocardiogram in our body, but this time is done on a few cells. And in this case, the beating was around 1 second per beat, which is the typical time for beating of cardiomyocytes. To show that indeed you are measuring the beating of the cells, you can add some drug, and this drug is, for instance, known to accelerate the beating of the cells. And indeed, when you introduce this drug, you see it beating a lot more, nearly double than when there is no drug in the system. So this shows that indeed this transistor configuration made of organic materials can be used to record the extracellular activity of cells. The important thing for us and why we choose this system is because this system is very stable over time. We have been able to record for weeks. So we can have the cells there for several weeks, and still the system is able to record the electrical activity. That's important because normally experiments are very long and you want to modify the properties of the system you want to analyze. And this is a system that ensures that you can do this type of an experiment. So this is almost at the end of what we have done, because now, of course, the last step, which is probably the most difficult one, is to couple everything together. So we want to couple this macroscale measuring system with the nanoscale measuring system, and in order to obtain from both macroscale properties and nanoscale properties. So this is working, which is under progress now. I hope to be able to update on this in some month, but this is probably the end of this type of technology that we want to develop. Okay, so with this, I can summarize the presentation. So the first message is you can use microscopic techniques to measure electrical properties of cells. And these are very versatile techniques, very powerful, but they lack the access to very small system or to the heterogeneity of the electrical properties of the system. If you want to improve this, you need to develop more powerful techniques like the scanning dielectric microscopy, which is the technique we have developed. And by using this technique now, you can have access to the local properties of the system, and you see there is a lot of heterogeneity, and probably this can reveal important information. And finally, the message is probably the best option is to combine microscale methods with nanoscale methods, because in this way you have the powerful of microscale and the powerful of the nanoscale together. We have done some progress toward this direction by using electrolyte gated transistors, and now we hope in some time to be able to reproduce these maps with cells sitting exactly on these transistors in order to have both macroscopic and nanoscopic properties of the cells. Well, to end up, of course, there's a lot of people I should acknowledge on this work, because this is represent work done over several years. Especially, I will acknowledge the work of Laura Fumagalli, with which we developed practically most of the techniques that I have explained today, and also several people which are now in my current research group. I cannot say all of them. Of course, also special acknowledgement to the collaborator group, basically biological groups or some specialized groups to get into different types of systems. And also, of course, thanking, acknowledging the funding from different sources, from the European Commission, the Spanish Ministry, or the Generalitat of Catalonia. And that's all. Thank you very much for your attention.